So, topic is molecularly targeted therapies for cancer. It's a very interesting topic. I told you the very idea of learning this course, molecular biology of cancer and so on, understanding the molecular biology of cancer helps you to find targets, molecular targets, because you know the conventional therapies you have some issues. You need novel targets. That is possible only if you have an idea about the molecular biology of cancer patients also. What are the molecules that are upregulated or down, whose expression is upregulated or downregulated, whether they are oncogenes, tumor suppressor genes, apoptotic regulators, DNA repair genes and so on. Those genes, how they are affected. If you have a complete profile of that, then probably you can come out with some therapy. So, that is how the therapies have been identified. So, conventional therapies as often repeated surgery, chemotherapy, radiation, side effects and lack of specificity are the major issues there. The other thing is the side effect is mainly because chemotherapy mostly acts on dividing cells. So, this inherently affects other rapidly dividing normal cells. We have discussed this. That is why you have these side effects like loss of hair, problems in gastrointestinal epithelium, bone marrow and so on. Nausea, vomiting, suppression of the immune system, renal and hepatic toxicity, hair loss and so on. Relapse and drug resistance are also major problems. The resistance to treatment comes from several mechanisms. Tumor cells may initially respond to chemotherapy, but after some time develop resistance to the same drug through several mechanisms. What are they? One is loss of importing of drugs. This is one. By this mechanism, the drug may be excluded by the tumor cells. So, that is one thing. There is another one, what is called multi-drug resistance. This is mainly due to overexpression of P-glycoprotein multi-drug efflux pump. This can effectively decrease the intracellular drug concentration of several drugs, not simple one drug. So, this is one mechanism. The other mechanism is loss of expression of drug metabolizing components such as cytochrome P450, so that the pro-drug will not be activated into an active drug. So, this is another thing. Inactivation of apoptotic proteins such as P53 by mutation or overexpression of anti-apoptotic proteins such as BCL2 will also, actually both will happen, inactivation of the pro-apoptotic genes as well as overexpression of the anti-apoptotic proteins, both may happen in one patient. So, increased inactivation of drugs or toxic intermediates by activating the relevant enzymes such as nucleoside deaminase. Alterations in the structure of drugs, drug targets such as this BCR-ABL, blood-brain barriers or inaccessible tissues such as testes. So, I have already told you what are the genes that are responsible for carcinogenesis, or what are the genes that are important in carcinogenesis, like these oncogenes, tumor suppressor genes, apoptosis regulators and DNA repair mechanisms and so on. These are further influenced by the microenvironment and stress signals, including the DNA damage. So, the subpopulation of cancer stem cells, all these things we are just recapitulating. I have already told you cancer stem cells may be important for initiating the tumor development. What do you mean by targeted therapy? Targeted therapy means the new generation of cancer drugs designed to interfere with a specific molecular target, usually a protein that is believed to have an important role in the tumor growth or progression. So, some protein which is important for tumor growth or progression can be an ideal target. So, that is the idea. So, this is why we need to have a detailed understanding of the molecular changes underlying cancer, then only we will be able to identify the appropriate targets. So, a protein becomes a druggable target if it contains a binding site, or maybe it is an enzyme, so it has a catalytic cleft or some enzymatic activity at least. Either structurally corner the catalytic cleft or just interfere with its activity. So, these are the ideal targets. Naturally, kinases are the very attractive targets. There are about 518 kinases in the human genome, out of which 90 are tyrosine kinases. So, tyrosine kinases are very important targets. Tyrosine kinase inhibitors, they form an important target. Transcription factors are thought to be not that much suitable targets except for hormone receptors, which are nuclear receptors, because the receptors can be on the membrane, receptors can be even inside some of them. One such example is some of the nuclear hormone receptors. So, such as estrogen receptor or progesterone receptors. So, they can be a good target, but otherwise transcription factors may not be a good target, because these receptors are also acting as transcription factors. That is why we are talking about it. Tamoxifen is a good example, and it modulates estrogen receptor alpha activity and is useful in treating breast cancer. The enzymatic activity present in RAS, what is the enzymatic activity? Do you remember? In RAS, it is a P21 molecule, yes. GTPase activity. Once the GTPase activity is there, what will happen to RAS? It will get inactivated. Once the GTPase is active, then GTP becomes GDP, and the GDP bound form is actually inactive. So, here if you should not inhibit the enzymatic activity, that is what I am trying to say, because the RAS by itself is a GTPase. So, when the GTPase is active, then actually it becomes inactive. RAS in relation to cancer, it is the mutation that we are interested in, not its enzymatic activity. That is the point there. So, thus the enzyme is not suitable as a target, since we should not inhibit this inhibitory activity. That is an important thing. It may be slightly confusing, but if you understand that RAS becomes oncogene by mutation, not by this GTPase thing. When GTPase is active, it actually becomes GDP bound form, which is inactive. That is what it means. Other thing is protein-protein interactions. Interaction sites are good targets, but it becomes inconvenient if there are too many points of contact between two proteins. It is not that only two proteins will interact with each other. That may be a chain reaction. So, if there are too many partners they interact with, then it becomes difficult to target only one of them or many of them. It is difficult that way. If you just target only one, perhaps the intended effect result may not be obtained. So, that is another. These are some just overview, but this is an important concept. Just a few minutes back, I was telling that you need several targets. In fact, cancer develops due to defects in multiple genes. So, it is a good idea to target many of them, but there are exceptions. One exception is called oncogene addiction. It is a very interesting concept, because It refers to a condition where the tumor cells are addicted to one particular gene, usually oncogene. So, the tumor maintenance is dependent upon the continued activity of this particular mutated or constitutively active oncogene. So, the whole thing depends on one gene. Can you tell one example? I have already told several examples in the class. HER2/neu. HER2/neu is an oncogene. In breast cancer, 20% of the breast cancer patients and ovarian cancer patients, this receptor, which is a second member of the EGF receptor family, is overexpressed. If it is overexpressed, it is a poor prognosis and there is an aggressive tumor growth. I have already told you. But if you stop this with a monoclonal antibody Herceptin that I told you repeatedly, then you are able to treat the situation. But you are only attacking one gene here, but you are curing the cancer, right? Or you are containing the cancer. That is what I am telling. Because the cells are addicted to that particular gene. The other example given here is the so-called BCR-ABL, MYC, RAS and BCR-ABL I already told you. So, these have been shown in some mouse models, oncogene specific, oncogene dependent tumor development in mouse models that has been demonstrated. Targeting of these oncogenes is generally done with a specific antibody or sometimes small molecule inhibitors, chemicals. Either you can inhibit them with chemicals if you have chemical inhibitors, or you can use some antibodies. Again, in antibodies, you should remember something. Antibodies do not mean all the time they will neutralize. What are called neutralizing antibodies? They will neutralize the effect, but there are also activating antibodies. There are many other antibodies. Simply antibodies do not mean that they will antagonize, because generally students think that way. So, it is not like that. Here generally we use antibodies that will neutralize that particular target. That is what we will use clinically for this kind of oncogene addiction, because we want to remove that molecule to which the cells are addicted. So, that is the idea. We will see the examples. This is a very interesting one. What is called BCR-ABL? I already told you, breakpoint cluster region. ABL means it is an oncogene, Abelson, because it was discovered by Abelson. There is a chromosome translocation. If you write the symbol like this, T922, it means the T stands for translocation. Translocation happens between the chromosomes 9 and 22. It generates what is called BCR-ABL oncogene. The fusion product is about 210 kilodaltons. That is why it is called P210. It is nothing but a tyrosine kinase. It is overexpressed in a particular type of cancer, which is chronic myelogenous leukemia. You can say blood cancer, CML. There are many different domains in the fusion protein. You may ask, why sir there is a fusion protein? BCR is one portion, ABL is one portion. They are joining together to form a fusion protein, which is a tyrosine kinase. There are many different domains of the fusion protein, and how this one will become a growth promoter. That is because this fusion protein, which is a tyrosine kinase, activates RAS signaling pathway, PI3 kinase AKT pathway, JAK-STAT pathway, all of which are known to be proliferative. So, it can give multiple, it can activate multiple signaling pathways, so that the proliferation takes place. It can also activate MYC, NF-kappa B, RAC, HCK is also another kinase, FES and JUN, etc. These interactions enable it to control proliferation and survival of NF-kappa B, survival, right. So, survival of cancer cells. So, this is an important point. I will show you how this BCR-ABL is formed. Let us look at it. Let us say this is normal chromosome 9. The blue portion is at the bottom is the ABL gene. It is present there. Let us say this is chromosome 22. I have given different color, and BCR is there. Now you see what happens in the so-called Philadelphia chromosome. Changes occur in both chromosomes. You see that ABL, a bit comes out, and BCR also broken. Now there is translocation. You will see that. See, in the CML, that is the chronic myelogenous leukemia, this is how the chromosome 9 looks like, and this is how the 22 chromosome looks like. So, see the changes. The blue one has acquired the red, and the red one acquired blue. So, changed chromosome 22 is what we call as Philadelphia chromosome. Although there are 9 and 22 both are getting exchanged, the Philadelphia chromosome generally refers to chromosome 22. That also you have to remember. Now, what is the mechanism and what is the drug? The drug is imatinib. Imatinib mesylate or Glivec was approved for treatment of chronic myeloid leukemia CML on May 10, 2001 by the FDA. This is what I told you, it was a very historic moment and people were very much excited because many of the patients were cured by this. So, basically imatinib not only inhibits BCR-ABL, it almost is equally very good or potent against other tyrosine kinases like platelet derived growth factor receptor, PDGF receptor, alpha, a particular one, and c-KIT receptor tyrosine kinase. And that is why it is also effective against other cancers which overexpress those receptors like gastrointestinal stromal tumors, GIST as we call, the most frequent mesenchymal tumors of gastrointestinal tract. And KIT mutations, KIT is also oncogene, c-KIT. Mutations are detected in about 75 to 85% of these gastrointestinal stromal tumors, while PDGF receptor mutations account for about 5 to 10%. So they are active against other cancers also. Now, let us look at what is this imatinib and how does it act, what is its mechanism of action. It is a competitive inhibitor of the BCR-ABL tyrosine kinase as well as these other tyrosine kinases, KIT and PDGF receptor, because what do they have in common? They are tyrosine kinases and they will use ATP for phosphorylating something. So, it is the ATP binding site to which this drug competes. That is why it is becoming a common site, because this ATP binding site is there in all the tyrosine kinases, these 3 tyrosine kinases. That is why it is not very specific for BCR-ABL, it is also acting on other tyrosine kinases. It competes with ATP for binding to the kinases, preventing the transfer of the gamma phosphate group to the suitable tyrosine residues. Thus, it is able to inhibit the downstream pathways. We will look at it. Now, let us say this is BCR-ABL and let us say the ATP, and then there must be a substrate which contains the tyrosine residue which needs to be phosphorylated. So, what happens? You have, let me put everything. See, when imatinib comes in, it replaces ATP. Otherwise, ATP will be binding here. Let us say this is the pocket to which ATP binds normally, and that will result in the elimination of ADP, and then the phosphate will be added to the some substrate, whatever that may be, right. So, this is what is the normal reaction, whereas now if you add imatinib, then it will not allow ATP to bind, it will competitively inhibit the ATP binding to the BCR-ABL, which is the enzyme. See, enzyme substrate, actually this is enzyme, the blue one is enzyme, this is substrate. Whatever it is phosphorylating, that must have a tyrosine residue which is phosphorylated. That is why it is called tyrosine kinase. And for that you need ATP, and ATP has to bind somewhere in the BCR-ABL, and that binding will be prevented in the presence of imatinib. So, this is a simple idea. If you have, that should be fine. See, when ATP goes, when imatinib binds, ATP is pushed out, just a cartoon. So, as I told you, there were several difficulties with this approach also. Patients have developed as many as 40 mutations have been found. So, people have generated several drugs, what are called second generation, third generation, and so on. So, some of them are listed here. You can take a look at that. Imatinib is the one that I just told you. Then this so called gefitinib, erlotinib, lapatinib, like that they have given some commercial names, sorafenib, abiraterone, and critozinib, vemurafenib. Whenever it ends with IB, I told you it must be a chemical. Whenever it ends with MAB, it is a monoclonal antibody. The name of the drug, just keep in mind. Like I told you, more than 40 mutant forms are known. So, the overexpression of BCR-ABL generally happens by amplification. Sometimes there may be upregulation of this drug efflux pumps and downregulation of drug influx pumps. There are several mechanisms. Alternative overexpression of how this resistance develops, that is what we are discussing. Sometimes there is some upregulation of drug efflux pumps, downregulation of drug influx pumps, and alternative overexpression of non-receptor tyrosine kinases such as LIN. Enrichment of CML stem cell progenitors that are resistant to BCR-ABL directed therapies. These are some of the mechanisms by which the cells can develop resistance to therapy. So, that is the idea. I already told you another oncogene addiction is by HER2/neu overexpression in breast and ovarian cancers, and this correlates with poor prognosis. Already mentioned this. So, this is another example for oncogene addiction. Here you can find that name Herceptin, which is called trastuzumab. This clearly tells you MAB is their monoclonal antibody to HER2/neu receptor. So, you want to know how it acts, the monoclonal antibody. It actually binds to the extracellular domain 4 of HER2/neu. If you remember, the HER2/neu structure is a receptor tyrosine kinase. All the receptor tyrosine kinases will have an extracellular domain, ligand binding domain. Of course, this particular one does not have a ligand, but it will have an extracellular domain, transmembrane domain, and an intracellular domain with tyrosine kinase activity, and a cytoplasmic tail where there are tyrosine residues to be phosphorylated. This is a typical structure of any receptor tyrosine kinase. So, here also. So, it binds to the trastuzumab, this antibody binds to the extracellular domain 4. There are several domains. The fourth domain is what it binds with. It is approved by the Federal Drug Administration for the treatment of HER2 positive metastatic breast cancer, and in the adjuvant setting in combination with chemotherapy. You can give in combination with other therapies, standard therapies. Trastuzumab downregulates HER2 signaling by masking a protease cleavage site on the extracellular domain 4. ECD means extracellular domain 4 and blocks shedding of the receptor, because ECD shedding constitutively activates the tyrosine kinase domain. You remember I told you there can be constitutive activation of the receptor, and this receptor can get activated either by simple over amplification or by this kind of cleavage of the extracellular domain. Part of it is removed, means it will get activated without ligand. So, that is one mechanism, and blocking this correlates with positive responses to therapy. And additional activity is also there through so called antibody dependent cell cytotoxicity, about which we will discuss in immunotherapy. We will later on talk about what is called the ADCC. We will talk about that at that time. Right now you understand that there are two mechanisms. One is simply blocking the extracellular domain, that is one thing, because it binds there. The other one is through antibody dependent cytotoxicity, cell cytotoxicity. Trastuzumab, the so called Herceptin, can also be used in HER2 overexpressing metastatic gastric and gastroesophageal junction adenocarcinoma. Why? Because that is also tyrosine kinase, right. So, this particular tyrosine kinase, here it is particularly against HER2/neu. Cardiomyopathy is one of the side effects. Sometimes you get there is no drug without any side effects, even this. Generally, the problem with the antibody therapy is it is a bit expensive compared to standard therapies. Antibody therapies are very expensive. Actually, Herceptin is being used in the clinics, even in Chennai. I know I have taken a patient and they are given Herceptin in Apollo, for example. So, it is widely used. The other thing is, in the same family of receptors, the so called EGF receptors, FDA has approved several antibodies. Some of them are listed here, monoclonal antibodies. HER1 means EGF receptor. HER means human EGF receptor 1. That means it is basically EGF receptor, epidermal growth factor receptor, right. So, the antibodies are cetuximab, panitumumab, a little bit difficult to pronounce, probably some Japanese name or something, but that is what it is, which disrupt autocrine loops in human malignancies, colon, head and neck cancer, and usually they are not standalone drugs. Standalone drugs means the drug alone is given, nothing else is given, but usually these are given along with chemotherapy. That is why I insisted on combination therapy. I explained a little bit on that in the last class. So, it is a combination therapy combined with chemotherapy, it is giving you higher response. Standalone also has a response, but in combination it works better. Several small molecule inhibitors also have received FDA approval, such as erlotinib, gefitinib. You remember gefitinib, I think I showed you with other BCR-ABL also, because it is tyrosine kinase inhibitor, here also it is working. Or somewhat HER1 specific, like lung and pancreatic cancer. Lapatinib is also a dual HER1 HER2 inhibitor, breast cancer resistant to trastuzumab. See here, they are using lapatinib, which is a dual HER1 HER2 inhibitor. If you remember the story I just told you, we have long back reported, it is actually my personal postdoc work, wherein I have reported that HER2 is a very good receptor. Even without ligand, it can heterodimerize with other family members and can take signals from the other ligands. So, that is why if you give, even if HER2/neu is a little bit less, then you can come out with this dual inhibitor. See, it is in the clinic now. It's all based on those kind of theories where we and others, not only ourselves, many others also have shown that it's a very good heterodimerizing partner. HER2/neu is a very good heterodimerizing partner. So, it can take signals from other family members. It won't take signals from any other receptor, only within that family. HER2, HER3, HER1, HER4 and so on. From those receptors, it can heterodimerize. But the signals can come from the so-called heterologous ligands. Heterologous ligands means the ligands are not directly to it, because it's an orphan receptor, it doesn't have a ligand, but takes up ligands of its sister receptors or whatever. So, the family members, they can be activated by cross phosphorylation. This also can be activated. So, that is the thing. That is why you may be able to, in some cases like lapatinib may be more effective. If the other receptors or if the other family members are also expressed, then it may be much more effective. So, such concepts which we have done in the lab are also finding their way into the clinic. That is what I am trying to emphasize. And naturally in research there will be so many groups working on the same problem and so on. Then another molecule is this PI3 kinase. Now we are away from this oncogene addiction. Now we are talking about other molecules, molecular targets, PI3 kinase, AKT. PI3 kinase are implicated in cancer because of the presence of activating somatic mutations in the catalytic subunit. See, PI3 kinase has two subunits, P110 and P85. So, the catalytic subunit is called P110, that are identified in approximately 30% of the epithelial cancers such as breast, colon, prostate, endometrial cancer and so on. Many agents have been discovered. We need not remember everything. About 10 P110 catalytic subunit targeted agents. Some commercial names are given, XL765, XL147 and so on. Some names are given. They have entered clinical trials. There is another oncogene, RET. RET also may come as sometimes, I do not know, maybe it is also coming under oncogene addiction sometimes, depends. But gain of function mutations in the RET proto-oncogenes leads to hereditary and sporadic medullary thyroid carcinoma, a rare calcitonin producing tumor arising from the cells, parafollicular C cells. These are called TKIs because they are called tyrosine kinase inhibitors. TKI means tyrosine kinase inhibitors. They are small molecules, chemicals. Basically, when we say, to distinguish them from proteins, proteins are macromolecules compared to because the molecular weight is, this is some 300, 200, something like that, whereas that one is in kilodaltons. So, to distinguish them, we usually call the chemicals as small molecules. So, like that. They are also targeting other RTKs. So, one advantage in this is that if other receptor tyrosine kinases are also responsible for the cancer development, then these kind of molecules could be effective because they are not specific for one tyrosine kinase, any tyrosine kinase, and that is useful. If they are multi-targeted, generally they are useful. That is what I told you already. Of course, single target mechanism comes when the cells are addicted to one oncogene. So, there you can use that. BRAF is another cytoplasmic. Here we are talking about serine threonine kinase that lies immediately downstream of RAS. If you remember RAS, RAF, MAP kinase pathway I have described, so you may remember that. It is a key player in the RAS MAP kinase pathway. This is especially very important one that you may have to remember. What is meant by this? V600E means what? The mutation is at the 600th amino acid, and the mutation that is happening is from valine to glutamic acid. These are single letter abbreviations. So, valine to glutamic acid, that is detected in melanoma, colorectal cancer, non-small cell lung cancer, and so on. I think this is parathyroid cancer, something like that. So, the so-called V600E BRAF mutation is a very important one. When that happens, it activates MAP kinase signaling. ERK means extracellular regulated kinase, which is again MAP kinase only. That induces proliferation and promotes transformation. So, the defect there is activation of RAS MAP kinase pathway, which induces proliferation. That is why BRAF mutation plays a very important role. Once you have this mutation, there is an activation of this pathway. So, such inhibitors are very, very important. This is very important to remember this inhibitor, which is called a sorafenib. The commercial name is Nexava or something. It is a multi-targeted kinase inhibitor that has activity against BRAF and also on VEGF receptor, vasculoendothelial growth factor receptor 2 and 3, and also on c-KIT, PDGF receptor, then that FLT3, and then FGF receptor, fibroblast growth factor receptor. So, these are all again tyrosine kinases. FDA again approved this for renal cell carcinoma and hepatocellular carcinoma, liver cancer and renal cancer. So, for in such condition, these drugs, this is a very famous one, sorafenib. It is approved and it is in the clinic. People are using that. Now, either you can inhibit the oncogenic proteins or enhance the tumor suppressor proteins. That could be another strategy, because the molecular targets, if you take as oncogenes and tumor suppressor genes are targets, let us say, the oncogenes means you will inhibit for treatment. Tumor suppressor means you will activate them. If there is a loss of tumor suppressor activity that happens in cancer, you want to activate, enhance the activity by somehow, whatever is the molecular mechanism that you know, then that could be a good strategy to treat cancer. That is what people are doing. So, generally we are not going to talk about epigenetic modifications now, but that is a major mechanism by which tumor suppressors are made inactive. The loss of expression of tumor suppressors is generally due to epigenetic modification. Epigenetic modifications are many. It can be DNA methylation, histone modification, and also the microRNAs. These are the three non-coding RNAs, you can say. So, these are the three things that will induce epigenetic modifications. We will talk about epigenetic modifications in detail. So, that is also one of the very interesting topics to discuss. That and DNA repair are very important mechanisms in cancer. So, we will be discussing them somewhat in detail. So, so naturally enhancing this kind of tumor suppressor activity could be done if you interfere with the p53 MDM2 interaction, because MDM2 inhibits p53. So, if you inhibit this inhibitor, you will be activating p53, simple, because p53 is getting degraded in the presence of MDM2, because through ubiquitin or it somehow that ligase E3, all those kind of things, mechanisms are there. So, it will help to degrade p53. You prevent that degradation by preventing the p53 and MDM2 interaction. That is done by another agent, which probably it is given also here, that is called nuclein 3, whether it's shown here or not, I don't know, but there are other agents. So, that is one important thing. Second thing is, since methylation and other things are involved, I told you now epigenetic alterations, sometimes you are using what is called hypomethylating agents, azacitidine and 5-aza-2 prime deoxycytidine, it is called decitabine, are approved for the treatment of myelodysplastic syndrome. There is another one which is a nucleoside analog, which is called Vidasa, the ribose structure that gets incorporated into RNA and DNA via ribonucleotide reductase. So, these are all some of the ones which can help in activating certain tumor suppressors. So, these are, we will look at the mechanisms later, right now you just understand. Similarly, what is HDAC? Histone deacetylase. So, histone deacetylase inhibitors also will be helpful. Later on, I will explain when I am explaining the epigenetic modifications, I will explain the details of the HDAC inhibitors and all that. For the time being, just understand, sometimes in tumors, HDACs favor deacetylation, because the name histone deacetylase. So, it favors deacetylation and tightening of histones leading to epigenetic silencing. So, this epigenetic silencing, see, silencing is a general word. If you silence an oncogene, it is good. If you silence a tumor suppressor, it is not good, because it will aggravate the cancer. So, it all depends on what is getting silenced. So, now you try to understand that there can be a dual role. Depending on these silencers, either microRNAs or epigenetic silencers or whatever, those which silence the gene expression ultimately may have dual role. It may activate, it may inhibit. Similarly, this acetylation, methylation, all these things can have a dual role in the activation of transcription or even inhibition of transcription, because it depends on what they silence. So, if you inhibit an inhibitor, you end up activating something. So, that is the mechanism. That is why this kind of contradictions come in. So, DNA methylation and histone deacetylation, they work together in gene silencing because of direct binding interactions between the so called DNA N-methyl transferases and HDACs. So, the methyl transferases and deacetylases, all these enzymes, you see, methyl transferase, demethylase or acetylation, deacetylation, you always have opposing reactions. All these are enzymatic reactions, and all these enzymes are actually together in the system in a complex. So, they interact with each other. So, the net result could be an activation or even inhibition. So, that has to be understood. Orinostat, this is very important. It is the first HDAC inhibitor to be approved for the treatment of T cell lymphoma. There are side effects like nausea, diarrhea, fatigue, anorexia and all that. It is basically this so called orinostat is nothing but suberoyl anilide hydroxamine acid. That is what they call sometimes SAHA, SAHA, suberoyl anilide hydroxamic acid. So, this is a very important compound. There is another one, belinostat, is also a novel HDAC 1, class 1 and class 2 histone deacetylase, and this class of compounds demonstrated anti-cancer activity in certain cancers like malignant mesothelioma and all that. I think we will stop here.